The digestive system is an amazing process filled with many different stages of development. These are you buying the food at the market, unpacking it, and then scoffing it down your throat. When you have bit into it using your front teeth and chewed it using your back teeth, you will then swallow it. Then it will travel down the esophagus or the gullet, as some people may prefer to call it. It will proceed to enter the stomach, where partial digestion of the food takes place. The churning action of the stomach muscles physically breaks down the food. The stomach releases acids and enzymes for the chemical breakdown of food. After food is churned up in your stomach, a sphincter muscle at the end of your stomach opens to squirt small amounts of food into the top of your small intestine. This first section of the small intestine is called the duodenum. Your pancreas releases digestive juices through the duct into your duodenum. This fluid is rich in enzymes that breaks down fats, proteins and carbohydrates. It also contains sodium bicarbonate, which neutralizes acid produced in your stomach. By the time food and digestive juices reach your large intestine, most digestion and nutrient absorption has already taken place. The large intestine's major jobs are to absorb excess water and to prepare feces or undigested waste material. Next up is the rectum. This is where the stinky stuff is stored, and then out it goes through the rectum, into the toilet, and into God knows where. Now we are going to talk about enzymes, and enzymes have baffled scientists for decades or even centuries now. These little things are positioned across the body and are crucial for our food to be digested. They can be called catalysts if you like, only biological catalysts. When in the digestion system, the food is absorbed by the tiny tentacles of the small intestine. Then, the nutrients go in the bloodstream, and the bloodstream carries it around the different cells so that they can function. The small intestine has small tentacles called villi, so that it can speed up its process and increase its surface area. Absorption is made possible by digestive enzymes, most of which are produced in the pancreas and released through the digestive tract. Our bodies produce enzymes specific to the type of food that we eat. Protein digestion enzymes such as protease, the fat digesting enzymes lipase and amylase for the digestion of carbohydrates. Food, when eaten, begins to digest in the mouth, but different types of food are digested in different parts of the digestive system. Proteins are primarily digested in the stomach. Fats are primarily digested in the small intestine. The process of the digestion of carbohydrates begins in the mouth with the release of the amylase enzyme, and then begin to break apart the bonds of the sugar altogether. Additional digestion of carbohydrates continues in the top half of the stomach. Protein digestive enzymes break down the bonds holding the amino acids together. All of these, particularly digested in carbohydrates, proteins, and non-digested fats, go into the small intestine. In the small intestine, amylase completes the process of carbohydrates. Fats are digested in the small intestine using lipase. These are then absorbed by the villi of the small intestine into the bloodstream. Enzymes are not only found in the digestive system, they are also found in cells. One of these enzymes is the catalase, which breaks down the hydrogen peroxide. The hydrogen peroxide then is broken down into water and oxygen. The substrate goes exactly into the enzyme. It breaks up and it leaves as a product. The enzyme can then be reused and reused and reused. Since scientists can't exactly zoom into an enzyme when it does one of its breakdowns, they have created a term called the lock and key theory. This is where a certain type of enzyme is adapted for different types of things. For example, lipase is specially adapted to fat, and when the two combine, magic happens. The enzyme, being the enzyme, contains an engraving, which is the active site where the substrate, being the fat, is inserted to. Perfect. It is then broken down into products, and then they leave. The enzyme may then be reused.